very much for giving me the chance to speak. And I think while I'm not going to talk about anything really neuroscience, my talk does follow Glenn's talk quite nicely in the fact that I'm going to be talking about venom still and how venoms could possibly help us treat diabetes. And particularly when we talk about cone snails and platypus. So you might ask, what on earth could these two organisms do to help us find treatments for diabetes? So our interests are in insulin and glucagon-like peptides. So glucagon-like peptide, or GLP-1, is an incretin hormone that increases concentration in the blood in response to food intake, and that leads to increased insulin secretion. So both insulin and GLP-1 are used in the clinic to treat diabetes. So the story today is going to be about how cone snail insulin could be potentially useful lead in our development of new insulins for treatment of diabetes, and how platypus venom is giving us a lead for novel GLP-1 treatments of diabetes. And the study that I've been talking about today is based on these two publications that now form the basis of the work we're doing in the lab at the moment. So just start with the insulin story. As you all know, insulin is secreted in response to food intake. So there's a rapid increase in insulin in the circulation in response to eating, and then there's a slower phase, and at times of no food intake there's a sort of basal level of insulin. And here you can see the insulin molecule. It's a two-chain peptide held together with 3 disulfide bonds. So it responds to intake of carbohydrate or glucose. So when you can't make insulin, if the pancreas and the islet cells can't make insulin, then you have type 1 diabetes. And that often occurs due to an autoimmune attack on the islet cells and so on. In the case of type 2 diabetes, you make insulin, but the target cells in the muscle and adipose can't respond to insulin. So they have the receptors on their surface, but the signalling inside the cell goes wrong, and that inhibits the intake of glucose into those cells. So in both cases, for type 1 and type 2 diabetes, glucose can't get into the cells and it stays in the blood, causing hypoglycemia, and you feel hungry and tired because your muscle cells don't get enough energy. Whereas too much insulin, and this might arise, say, even in diabetes, if somebody oversteps the mark with their insulin treatment, then it leads to low blood glucose or hypoglycemia, and you get clumsy, slow and confused. And this point will become important in a little while. So I don't really need to tell you much about diabetes and the fact that it's a huge problem in the world at the moment, and of course most diabetes relates to, 85% relates to type 2 diabetes. It's an enormous personal cost and health cost. There are a huge number of problems arising due to long-term diabetic complications listed here, and so that really is something that is highly desirable to avoid, and it arises due to fluctuations in blood glucose over time. So there are good treatments for diabetes, of course, and a lot has been done over the time to make new analogues of insulin, which really help to emulate that biphasic insulin response. And listed here are some very commonly used insulins in the clinic. And here you can see the mutations that have been introduced into the insulin molecules that give them their specific properties. So there are so-called short-acting insulins and long-acting insulins. Short-acting ones emulate the immediate postprandial insulin response. And what is happening in these cases is that the insulin stays in its monomeric form rather than the stored hexameric form. It doesn't form dimers and hexamers. And that's due to interrupting an interaction between the end of the B chain here between two molecules. So most of these changes are at the end of the B chain. The long-acting insulins emulate basal insulin and stay around a lot longer due to changes in their properties, change in PI, for instance. So what's wrong with those? Well, one thing that we could improve upon is the initial response to blood glucose. So this first phase, the short-acting insulins are very good, but they're just not rapid enough in a lot of cases. So it would be great to be able to improve this. So this is where cone snail come in. So why cone snails? So first of all, to introduce cone snails, shown up on the top right hand here. They're quite large snails found in tropical waters, and they all produce venoms. Many of them have a mechanism which shoots a harpoon out to kill their prey, but some have a different mechanism, which I'll come to in a minute. So how did we come to looking at cone snails in terms of insulin? Well, my collaborators, Helena Safavi and others, Danny Chu and Ray Norton, were doing transcriptome analysis on the venom and found that indeed the venom contained insulin-like peptides. This was quite unexpected. So from this we thought, well, and a lot of my work is based on this sort of hypothesis at the moment, that if we can understand the structure and function of insulin-like peptides from a diverse range of species, this will inform us of novel mechanisms of the interaction with the receptor, and therefore maybe allow us to develop insulins with new properties. Okay, so here we have the cone snail insulin, that Helena was working on, and it's a particular. She was working on a couple of species that actually have a different mechanism of capturing prey, and that is to extrude a whole lot of venom over the prey, and the venom itself acts to slow down the prey and allow the cone snail to capture it. So here you can see that a couple of species that she found that have these properties, including this Conus geographus. And it turns out that the cone snail insulins that these guys produce have this particular property that will slow down the swimming of that prey and effectively induce a hypoglycemia in the prey. So this sparked us to look at the actual insulin molecules, and here you can see the sequences of cone snail insulin compared to zebrafish and human insulin. And the first thing we could see was that in fact the sequences are very similar to fish insulins, in particular parts of the molecules that are important for binding to the receptor. So a lot of my work has been in the past understanding how insulins and IGFs interact with their receptors, doing lots of mutagenesis and so on. So we know a lot about which parts of the molecules are involved in that interaction. The second thing, so it looks like the venom, cone snail venom insulin has evolved to work well in fish, which is quite sneaky. And then the next thing we notice there are some sequence differences in particular parts that are interesting. Also, as Glenn alluded to, there are very interesting peptides in venoms, and often they have really interesting post-translational modifications. So here you can see the cone snail insulins have 3 post-translational modified residues, which we never see in mammalian species. Okay, and the third most striking thing we saw was that the cone snail insulin lacked this end of the B chain here. So I told you earlier on that that end of the B chain was very important for dimerization, but actually its major function and is to be the primary anchor point for insulin on its receptor. So we were thinking, well, this is really weird because how could it actually be active on the insulin receptor? So this is where our work came into play, and we looked at the actual receptor binding of the insulins to see if they could indeed bind the insulin receptor and how potently. And we compared that to human insulin. So here you have a competition binding assay with human insulin and the cone snail insulin. We also had a version which doesn't have those post-translational modifications. It worked a little bit worse, so they do play a role in the binding. So what I don't have here is the human insulin that lacks the 8 amino acids at the end of that B chain. If we did, it would be right out here. So funnily enough, this insulin can bind with really quite good affinity, because if we did the same truncation in human insulin, it doesn't bind at all well. So that was very surprising. And also we showed that they were active in promoting downstream signalling via AKT signalling here. So this was really a remarkable finding, that the insulin now must be binding to the insulin receptor in a different way to human insulin. So this provided us then with a real template for making quite novel insulins and with unique properties. So we went along with Mike Lawrence to determine the structure of the cone snail insulins to try and understand what the mechanism is. And here we have them overlaid with human insulin. So here we have the so-called A chain and the B chain. The black is human insulin and the blue is cone snail insulin. You can see they're almost identical except for the lack of the end of the B chain. So we couldn't work out why they would be different based on the structure, but then we did modelling with Brian Smith at La Trobe to molecular dynamics simulations to see how that insulin might sit on the receptor structure. So we had Mike Lawrence had determined the structure of part of the receptor with that primary binding site that I was mentioning. Here mapped on the structure, there's a sort of surface that the insulin lies upon in white. The black residue here is actually human insulin locking onto the surface of that insulin receptor. And we can see that the molecular dynamics told us that this tyrosine at 15 was substituting for that tyrosine that wasn't present anymore. So it has a different way of grabbing onto the receptor. So we're now at a state where we've taken all this information and tried to make a whole lot of human variants that bind to the receptor based on this mechanism rather than the original mechanism or the normal mechanism. And the key point is that these new insulins will be very rapid acting because they don't form the dimers anymore. So they don't have to undergo dissociation and so on. They can just bind directly to the receptor and be very fast acting. So we believe we have some very interesting molecules which are currently in preclinical trials going on to be commercialised in Texas. And also we're doing a whole lot more work on a whole lot of other species of cone snail and coming up with other mechanisms of binding which look really interesting. So that's where that story is. And now I'll turn to the GLP-1 story, which is looking at monotremes. And this work has been done with Frank Grutzner at Genetics Department at Adelaide Uni, and it's a collaboration between Monash Uni, Adelaide Uni, and is supported by MedVet Science. So Frank came to me one day because he had been involved in the genome project that sequenced the platypus genome, and they had some very interesting things that they could find there. So why look at monotremes? We like to look at monotremes because they're the first emerging mammal mammalian species 166 million years ago. So that gives you a really good snapshot of the evolution of all sorts of processes. So we wanted to look at evolution of metabolic processes. They also noticed that the stomach's very small and lacks, is a high pH, and the genome showed that there were a lack of many genes involved in digestion, involved in digestion in the stomach. So that suggested we might look at metabolic genes, and that from there we've looked at things like ghrelin, which isn't present in the in both platypus and echidna, and we've looked at the architecture of the islets. It's a little bit different. So we looked closer at insulin and insulin controlling peptides. So GLP was one. We already knew that it was a pretty interesting molecule for other reasons. This has gone a little bit weird, but here you can see human GLP-1 encoded by the glucagon gene. It has a particular residue here at position 8, which is a target for cleavage by the enzyme DPP4, and this allows GLP-1 to be very tightly regulated. So insulin release is very tightly regulated. But interestingly, the platypus and echidna sequences that we pulled out had changes at this position, which was reminiscent of the story from the Gila monster venom sequence, which had a change at this position and and this peptide has gone on to be in the clinic as a very popular treatment for type 2 diabetes. So we have now seen that the platypus and echidna GLP-1 have this interesting property. We saw that it was expressed not only in the intestine where we expected, but in the venom, as I alluded to before. But the interesting evolutionary perspective is that there's only a single GLP-1 gene in the monotremes. The same gene is expressed in the intestine as the venom, whereas in the Gila monster, which is where Extendin 4 comes from, there's an endogenous normal GLP-1 and a venom GLP-1, which has evolved to be protease resistant. So it's quite a different system. So we looked at protease resistance and showed that, in fact, the monotreme molecules were resistant to DPP4 cleavage. And so it's an ultra-stable form, and we think it has great potential as a new type 2 diabetes treatment. Interestingly, it is degraded in its own serum, and there obviously has evolved a different way of inhibiting its action in monotremes, but that's something we would like to understand better in the future. We've also looked at its binding to the receptors, the human and platypus GLP-1 receptor. Interestingly, it's a pretty low affinity interaction. Excuse me. But it does stimulate insulin release in rodent islets. But interestingly, it's showing different signalling bias. It actually promotes signalling by ERK1 and 2 much stronger than we would expect, excuse me, compared to the other signalling pathways. So what is next? We want to look at monotreme GLP-1 and see in vivo and see if it really does act to lower blood glucose in vivo. And does it does the signalling bias towards ERK1 and 2 give us additional benefits than, thank you very much, I just couldn't make it to the end, than others, thank you. So does it actually, so we're looking at other endpoints such as gastric emptying and non-alcoholic fatty liver disease to see if this molecule will have actually different properties to human insulin and give us an edge there. So the take-home message is comparing genes and proteins from different species has provided us with unique clues about how we can build novel diabetes treatments in the future. And I'm even turning towards Drosophila now, so I'm adding species as we go. So thank you very much, and to acknowledge all the workers in my lab, my collaborators at Adelaide Monash, Utah, etc., and MedVet for their support, and other funding sources for their support along the way. Thank you.